grateful to the organizers for their invitation to be part of this wonderful symposium. I am very honored to be here today. This work I'm going to talk about is a study I conducted at AIST in my previous position. I worked with great collaborator, especially Dr. Nobu, who is my supervisor when I was postdoc at AIST, and the study has already been published. In this talk, I focus on how genomic data allow us to trace not only common ancestry, but also the evolutionary processes that generated diversity and individuality among phototrophs. If you're interested, I'd be happy to share the paper with you. So, firstly, I like to trace back the Earth's history. The Earth was formed approximately 4.6 billion years ago. At its birth, the planet was surrounded by hot and unstable environments, and it was far too harsh for life to exist. However, now Earth has diverse ecosystems with the diverse organisms, including plants and animals. The question is, how do we trace the evolutionary history of life? Geological record is a natural archive that preserves information about Earth's history of life over a past period of time. Examples of geological records are fossils, isotopic signatures, and molecular biomarker like lipid from cells. Here is a beautiful 2.7 billion years old fossils of photosynthetic organisms. Fossils like these provide past environment conditions such as atmospheric conditions including oxygen and also past biological activity. Then let's check the timing of key biological events revealed by geological record. This is a diagram of the age of the Earth's history with a scale up to 4.5 billion years ago when the Earth was formed. From the isotopic evidence, it is considered that the earliest life emerged at least 4.1 billion years ago. Fossil evidence suggests that photosynthesis already exists around 3.5 billion years ago, and eukaryotes around 1.8. Before eukaryotes, oxygenic phototrophs were thought to have evolved. The blue line provides the oxygen concentration on Earth. The Earth was firstly surrounded by an anoxic atmosphere with no oxygen. But after oxygenic phototrophs appeared, oxygen started to accumulate on Earth. This changed the atmosphere from anoxic to oxic and led to the diversification of organisms, including eukaryotes. How oxygenic phototrophs evolved? Oxygenic phototrophs are restricted to cyanobacteria among prokaryotes and have evolved later than anoxygenic phototrophs. Early phototrophs used, for example, reduced sulfur compounds, and they did not produce oxygen, but oxygenic phototrophs use water and release oxygen. Geological records give us information about the timing of their existence, but they don't explain how this metabolic system evolved. So, to understand the evolutionary processes, we need to rely on genomic and phylogenetic analysis. In this slide, I show how genomic information helps us understand evolutionary processes. First, by comparing genome or protein sequences, we can cluster organisms based on sequence similarity. This allows us to identify groups of related species or proteins. Using these similarities, we can construct phylogenetic trees which represent evolutionary relationship among species, proteins, or metabolic functions. These trees trace back to common ancestral cell and illustrate how distinct lineages diverge over time. Importantly, phylogenetic tree can also be used to estimate divergence times, providing a temporal framework for evolutionary events. So, tracking evolutionary history of the function, phylogenetic analysis is necessary. To apply this approach to photosynthesis, let's look at which proteins are involved in photosynthesis in next slide. At its core, photosynthesis requires two key components, light-absorbing pigments and reaction center protein, where photochemical reactions occur. On the left, I show anoxygenic phototrophs. These organisms used bacterial chlorophyll, showing the solid line here, as their pigments and possess either type 2 or type 1 reaction centers, but not both. And they don't produce oxygen. On the right are oxygenic phototrophs, such as cyanobacteria. They use chlorophyll A and uniquely contain both type 2 and type 1 reaction centers, known as photosystem 2 and photosystem 1. Are there any relationship between the phylogenetic distribution of bacteria and phototrophs? Here is the bacteria phylogeny showing phototrophs lineages and the type of reaction centers they possess. As you can see, phototrophs are sporadically distributed across the bacteria phylogeny, making it difficult to identify a single common evolutionary patterns. Recently, the number of culture strains showing star has increased, but it still scatters phylogenetic distribution, and it raises a fundamental questions. How can we better understand the evolution of photosynthesis? Recent breakthroughs in genome analysis provide a powerful opportunity to revisit this problem. Sequencing costs have dropped dramatically by nearly 1,000,000-fold over the past 20 years. At the same time, genome databases have expanded rapidly, with the number of bacteria species increasing almost 5-fold in the last 5 years. These developments allow us to revisit phylogenetic analysis at an unprecedented scale, providing much higher resolution for understanding the evolution of photosynthesis. So the research question is tracing the evolutionary history of phototrophs with phylogenetic analysis using more than 10,000 genomes. And try to answer the question we did. Try to answer the question. So we have 3 questions. Do we trace the ancestral photosynthetic organisms? How did extant forms of photosynthesis arise? And when and how did oxygenic photosynthesis evolve? Here is the method. First, we constructed a robust bacterial species tree using more than 10,000 genomes representing overall bacterial evolution. In parallel, we constructed a phylogenetic tree for photosynthesis-related proteins, including reaction center proteins and pigment biosynthesis enzyme using more than 40 genes in total, but I will not show all of the results today. And We then compare the topology of the species tree and the proteins trees. If the evolutionary history of photosynthesis-related proteins is congruent with the species tree, this suggests a species obtain the protein via vertical transmission. In contrast, incongruence between these trees indicate horizontal gene transfer. To ensure reliable tree topology, we applied multiple routing and reconciliation approaches, including outgroup routing, duplication, transfer loss reconciliation, and the minimal ancestor deviation method. Here are the first results, revisiting a phylogenetic tree of bacteria collapsing with the phylum. Then mapped groups that contain phototrophs onto the phylogeny. Our updated tree showed 4 phototrophs are gathered in one group. Then we found some hint at the ancient traceable link between phototrophy within this lineage. This figure shows a simplified bacterial phylogeny, highlighting the so-called Terrabacteria group. In this group, 4 phyla of phototrophs are relatively clustered, showing star. Within this group, Chloroflexi, Eremiobacteriota, and Firmicutes have a bacterial chlorophyll based on photosynthesis. In contrast, cyanobacteria uniquely use chlorophyll for photosynthesis. So we observe a basal split here between bacterial chlorophyll associated and chlorophyll associated lineages. This pattern suggests that the divergence between bacterial chlorophyll and chlorophyll synthesis occurred early in evolution like here. So to test this hypothesis, we compare the species tree on the left with the gene tree on the right, focusing on bacterial or chlorophyll synthesis. These enzymes catalyze the final and committing step of bacterial chlorophyll or chlorophyll synthesis. In the gene tree, we observe a clear separation between 2 groups, BCHG, which is associated with bacterial chlorophyll synthesis, and CHLG, which is associated with chlorophyll synthesis. This indicates that the divergence between bacterial chlorophyll and chlorophyll synthesis is deeply rooted in evolution and reflects an early functional differentiation rather than recent horizontal gene transfer. To evaluate whether the observed gene tree topology can be explained by vertical inheritance alone, we apply DTL model that explicitly accounts for gene duplication, horizontal transfer, and gene loss events. This approach allows us to reconcile the gene tree with the species tree and to infer the most likely evolutionary scenarios. By integrating the species tree and gene tree and DTL-based reconciliation, we infer that BCHG and CHLG began diverging from a common ancestor within Terrabacterium, showing here. Pink highlighted is showing vertical inheritance. This divergence appeared to have occurred early, followed by vertical inheritance into at least 4 major lineages, including Chloroflexi, Eremiobacteriota, Firmicutes, and cyanobacteria. In contrast, outside Terrabacterial group, bacterial or chlorophyll synthesis do not follow the species phylogeny. Instead, their distribution is best explained by horizontal gene transfer events. Taken together, these results suggest that the core divergence between bacterial chlorophyll and chlorophyll synthesis originated within Terrabacteria with subsequent vertical inheritance and later horizontal spread to other bacterial lineages. Based on this model, we next examined how reaction center proteins evolved in related to pigment synthesis. Importantly, a similar evolutionary pattern is observed for reaction center proteins. Both type 1 and type 2 reaction centers show a basal divergence from a last common ancestor shown here, followed predominantly by vertical inheritance within Terrabacteria. These parallel evolutionary patterns suggest that pigment diversification and reaction center diversification were tightly coupled early in evolution. From those results, an ancient Terrabacterium likely possessed prototype version of all extant photosynthetic machineries, such as pigment synthesis and reaction center, providing the foundation for the later diversification of phototrophs. So we define the last common ancestor of all photosynthetic organisms as LAPCA. And next, we try to find out what other metabolic functions LAPCA might have had. Since photosynthesis and carbon fixation are tightly coupled in modern phototrophs, we focused on CO2 fixation, specifically the carbene cycle. We reconstructed phylogeny of key CBB cycle marker proteins, including phosphorubilokinase and type 1 rubisco large subunit, and compared them with the phylogeny of phototrophic lineages. As shown by the blue and green clays, which represent vertical inheritance, the phylogenies of these carbon fixation proteins reveal the same major phylogenetic division as photosynthetic lineages within Terrabacteria, tracing back to LAPCA. So LAPCA was not only photosynthetic, but likely capable of autotrophic CO2 fixation as well. Let me summarize our first two questions. Do we trace the ancestor of photosynthetic organisms? The answer is yes. We trace last common ancestor of all photosynthetic organisms, showing yellow star. And how did extant forms of photosynthesis or phototrophs arise? We found Terrabacteria vertically and others horizontally acquired photosynthesis. Next question is, when did they evolve? To answer the question, we did molecular clock analysis. Molecular clock analysis is a tool used to estimate the timeline of the tree of life. In this study, we used a genome-based species tree including bacteria, archaea, and eukaryotes to estimate divergence time. We then estimated divergence time using BEAST2 software calibrated by well-established geological and fossil constraints as listed here. Because bacteria fossils are very limited, eukaryote fossils like plants, animals, algae are essential for constraining divergence times in molecular clock analysis. Here is the results for time estimation. Tree branch lengths are adjusted to the geological time scale, and yellow bar indicate the error bar of the divergence state, and red circle showed calibration points. Here is eukaryotic clade and archaeal clade and bacterial clade here. To see the branch of diversification, divergence time of Lapka is estimated to have emerged at least 2.7 billion years ago. And the next question is, does Lapka have oxygenic photosynthetic machinery? To address the question of whether Lapka already had oxygenic photosynthetic machinery, we focus on type 2 photosystem, which is essential for oxygen evolution. First, it is important to note that type 2 reaction center proteins, PAF, LM in anoxygenic phototrophs, and PSP-AD in oxygenic phototrophs are homologous proteins derived from a common ancestral reaction center. This indicates that Lapka already possessed a primitive type 2 reaction center, and a question is whether it possessed the ability to perform oxygenic phototrophs or not. The key innovation of oxygenic photosynthesis lies in PSP-A proteins, which bind the metal cluster responsible for water splitting in heteromeric PSP-AD subunits. So the residue for metal cluster is important for generate oxygen. So we examined whether the residue were already present in Lapka using ancestral sequence reconstruction of PSP-AD. Ancestral sequence reconstruction showed that residues required for metal cluster binding were not present in Lapka, but preserved at common ancestor of PSP-A and D, showing here. It was suggested that the protein capable of oxygen generation was completed here after speciated with anoxygenic phototrophs reaction center proteins. We did another molecular clock analysis for this clade. This showed that the divergence time of this clade is at least 2.4 billion years ago. So our molecular clock analysis indicates that oxygenic phototrophs evolved later Lapka around this age, and this timing matched the great oxidation events, providing the first molecular evidence directly linking oxygenic phototrophs to the rise of atmospheric oxygen. This leads to an important question. Did the oxygen-evolving ancestral protein already exist before cyanobacteria? To address this, we checked when cyanobacteria themselves emerged. So we back to molecular clock analysis again. To see here, cyanobacteria emerged relatively late, around 1.5 to 0.93 billion years ago. This suggests that the core oxygenic photosynthetic machinery predates the crown group of cyanobacteria and was already present before their diversification. Putting these results together, extant oxygenic phototrophic cyanobacteria evolved later than the first oxygenic phototrophs. This means that the first oxygenic phototroph was not an extant cyanobacteria, but rather an ancestor of cyanobacteria. In summary, Lapka was are non-oxygenic carbon-fixing phototrophs, and oxygenic photosynthesis emerge near the great oxidation events, and extant cyanobacteria evolve later than the first oxygenic phototrophs. It is important to note that LAPCA is not the origin of photosynthesis, but the last common ancestor that we can reconstruct and trace using phylogenetic data. Geological records suggest that photosynthesis predates LAPCA, indicating a gap that likely corresponds to extinct or unsampled early photosynthetic phototrophic lineages. This raises as a question: are there ancestral phototrophs molecular evidence at 3.5 billion years ago? And if so, which proteins can trace that deep past? We found some good marker proteins which is involved in pigment biosynthesis. The biosynthesis pathway for photosynthetic pigments is shown here. It derives from central metabolisms and produce chlorophyll or bacterial chlorophyll. The synthesis of chlorophyll pigments proceeds by the insertion of magnesium into protopropylene. In this context, we have focused on the core proteins BCH and BYZ. This complex is involved in the synthesis of precursor of the photosynthetic pigments chlorophyll A and bacterial chlorophyll A, which is responsible for reduction of tetrapyrrole at specific site, showing red circle here, respectively. The four proteins BCH and BYZ are belonging to the same protein family, but phylogenetic relationship was unknown. This is because of these four proteins alone are not sufficient to reveal their phylogenetic relationship. They have homologue, but it is too far to calculate. So we try to reveal the phylogenetic relationship of the protein family using large data and estimate divergence state again. Here is the phylogenetic tree constructed using approx 10,000 sequences from the protein family that includes core pigment synthesis proteins. The gray branches represent proteins with unknown functions. By constructing a large-scale phylogenetic tree that includes these previously uncharacterized proteins, we were able to resolve the detailed topology of this family protein for the first time. Importantly, we show that all photosynthetic pigments protein we use here diverge from a single common ancestral proteins. From the divergence time estimation, we found BCH and B emerged at least 2.3 billion years ago, and BCHYZ emerged 2.0 billion years ago, and found the upstream enzyme BCH&B appeared earlier than the downstream enzyme. This is a reasonable result considering the evolution of biosynthetic pathways. Are extant enzymes used in ancient period? Through the reconstructed tree, we found that those enzymes have a common ancestor here alpha and beta, then we could trace back photosynthesis to at least 3.7 billion years ago. There are reports that modern enzymes have more specific reactivity than their ancestor CFPD. CFPD is the most ancient proteins in the family tree, and it can reduce multiple reaction site and less specificity. And BCHYZ also could mediate reduction of multiple sites in tetrapyrrole sometime. So ancestral protein catalyze multiple reduction reactions with less specificity and generates a precursor of BCH bacterial chlorophyll A and maybe precursor of chlorophyll A simultaneously as well. Who have an ancestral form? Common ancestor of BCH and BYZ have emerged earlier than bacterial diversification. This point is the last common ancestor of bacteria. And as a molecular evidence, we tried are untraceable. So the summary, all this molecular evidence of photosynthesis, we found that photosynthesis, and we could trace back photosynthesis to 3.7 billion years ago, but only pigment biosynthetic origin, biosynthesis proteins. And origin of photosynthesis is probably ancestor of bacteria, but have lost multiple lineages. And here is take-home message. By combining phylogenetic analysis with geological records, we can enhance the resolution of when and how life evolved. And what we did was revealing what evolutionary history is traceable and untraceable using molecular evidence. Future observation we want to try is estimating the physiology of ancient photosynthetic bacteria. Thank you for your attention.